So the next thing we did is to take estrogen receptor alpha knockout mice. And the first thing we wanted to do is to, this is actually an in vivo study, and these are ovofrectomized female rodents, either wild type, and when wild type is stimulated with estradiol, and then we look at the hypothalamus, there's an increase in progesterone production in the hypothalamus, which goes along with the LH surge. But in estrogen receptor alpha knockout mice, there was no increase in progesterone. In the literature, the estrogen receptor alpha knockout mice actually don't have positive estrogen feedback, they don't have LH surges, and they're not sexual, they don't have sexual receptivity. The lordosis quotient is almost 0. So estrogen receptor alpha is very critical for estrogen positive feedback and for sexual reproduction.
John, and I'm not aware, do these alcohol ovulate at all?
No.
They don't have spontaneous?
No, they don't have estrogen positive feedback, they never have an LH surge. So
but if you give them exogenously to ovulate, with gonadotropin, do they ovulate? I'm just trying to think whether it's a central mechanism or a peripheral, just for my curiosity.
I don't know if there's any studies on giving gonadotropins. I think it's more of a central mechanism, but I don't know if it's also in the ovary. I just know that there's no estrogen positive feedback, so it's never a trigger. But that's interesting. So this is consistent that the wild types can have a progesterone response after estradiol injection, but not the ERCOs. And so in vitro, we took astrocyte cultures of the wild types and compared them to the ERCOs. And we see an estradiol will stimulate a calcium response, but in the ERCOs, this calcium response is attenuated, and the calcium response was not significantly increased. So this tells us that estradiol mainly signals through probably the estrogen receptor alpha. Also interesting was the DPN at 1 nanomolars, again signaled a little bit in the wild types and a lot more at 100 nanomolars, but this was actually abolished by estrogen receptor alpha knockouts. So therefore we think that, again, the DPN is not a pure estrogen receptor beta agonist. It actually, it's about 70-fold higher preference for estrogen receptor beta. So at 100 nanomolars, it can actually also activate estrogen receptor alpha at about a 1 nanomolar response. So that's probably what's happening here, is that estrogen receptor alpha is being activated by DPN and not beta. And STX and G1 are actually fully able to signal through estrogen receptor alpha knockout mice. So this demonstrates a few things. One, it demonstrates that there's probably a different signaling mechanism. It's independent of estrogen receptor alpha. But also, two, that even though this signals and it has a very high calcium level, it may not be relevant in reproduction because these mice actually don't produce progesterone and they don't ovulate. So summary from the ERCO mice is that the estradiol-induced calcium release was abolished, which supports estradiol signaling through membrane estrogen receptor alpha. And it's consistent with lack of estrogen-positive feedback and lack of sexual receptivity in ERCO mice. DPN at 100 nanomolars did not stimulate the calcium release, which suggests that DPN acts upon membrane-associated estrogen receptor alpha and not membrane-associated estrogen receptor beta. STX induced a similar calcium release compared to the wild types, which suggests that the signaling pathway is independent of MER alpha, but it's of unclear physiological relevance and function. And then G1 induced calcium release, which was not blocked, so that's still consistent with our hypothesis that it could signal on the plasma membrane for mGluR1A, as well as activate directly intracellular GPR30. So overall, we believe that membrane estrogen receptor alpha is the main receptor responsible for rapid estrogen signaling in hypothalamic astrocytes that's involved in estrogen positive feedback and the LH surge. So the last part of the talk is going to focus on sex differences in hypothalamic astrocytes. And if we compare male and female astrocyte cultures, the females that we've been studying so far respond with a calcium increase. The males actually also increase calcium, but it's at a lower level. So that's interesting that the males can respond. However, that when we look at the progesterone, we use 1 nanomolar, and the female will release and synthesize and release progesterone, but the male mice and male rats actually don't increase progesterone production. And then we tried it with 100 nanomolars, and again, the estrogen can induce progesterone synthesis in females, but not in the males. So again, this may also tell us that there may be some threshold that this lower calcium may not be enough to stimulate progesterone production. But it's unclear if it's, if these mice had a high enough calcium, whether or not they would produce progesterone. So that would be a study. So I just wanted to talk briefly about surface bioattenuation because one of my studies involves this process. So there's commercially available kit, sulfonyl monophenol NHS biotin, and the biotin is a large molecule. It's water soluble, but it's membrane impermeable. And with this, you can actually cause reactions to primary amines, either on amino acids or on N-terminus, if they stick out of the plasma membrane. So anything that sticks out of the plasma membrane, this can form amide bonds if it has a primary amines. And the purpose is to demonstrate if membrane estrogen receptor alpha has an extracellular portion that sticks out of the membrane, and also it can tell us if estradiol levels can actually regulate the insertion and internalization of membrane estrogen receptor alpha. So I'm actually not going to talk about the internalization studies, because that kind of goes off my talk, but I'm going to just talk about insertion really fast. So if you have an intact cell, you have some membrane, in this case estrogen receptor alpha, and then you incubate with estradiol. We want to see if this would actually cause an increase or decrease of membrane estrogen receptor alpha levels on the membrane. Then, after incubating, we would biotinylate, and then which forms stable amide bonds, and then we want to wash off the excess free biotin. And then we take these cells, we lyse them, run them on a column that recognizes the biotin, so anything that is sticking to the biotin is on the column, and then we elute the column, run it on the Western. So this is not specific binding, it could be any protein or membrane receptor on the plasma membrane, but then we probe this for our membrane receptor of interest, which is estrogen receptor alpha. So I participated in the study. This is not my primary study, but we can actually see that at about 30 minutes of estradiol exposure, that there's actually increase in estrogen receptor alpha concentrations on the plasma membrane, as well as an immunoreactive, which we believe is the splice variant of estrogen receptor alpha. Actually, and they both increase in parallel, and then decrease, and at 24 to 48 hours, it's actually back at baseline levels. So this tells us that the estrogen is regulating the membrane estrogen receptor levels on the plasma membrane. And I wanted to talk about that because we actually did an experiment. We know at 30 minutes, there's the maximum amount of estrogen receptor on the plasma membrane. So we looked at males and females at control, and we see that males and females both have estrogen receptor alpha on the membrane, and the females were not significantly higher than the males. And then after about 30 minutes of estrogen exposure, in the females, the amount of membrane estrogen receptor increased, which was statistically significant. The males' increase was not statistically significant. So this tells us that maybe this is some mechanism where there may be more response of estrogen receptor alpha going to the membrane, and maybe being able to generate a higher calcium response.
Yeah, did you see with the other agents which release this calcium, whether there's a difference in the male and female?
We actually didn't study all the other compounds. We didn't study the other agonists in the males.
No, because one you talk of receptors, but it may be a fundamental difference that they just don't release more calcium, right?
Right.
So
It's
that
possible.
would be, yeah.
Yeah, it may not be a threshold, it may be some different.
Yeah.
I mean, John, just to add on that, I know at least in human GVO site, androstenedione antagonizes the ability of estradiol to mount calcium flux, and I'm wondering, are some of these sexual dimorphic changes due to androgens?
You mean like earlier on in development and it changes the cell?
Well,
Or-
perhaps, but even acutely. In other words, if you co-incubate a GVO site with estradiol and you induce these calcium oscillations, you can block them with androstenedione. So you could actually posit an acute effect as well.
Well, these are all in culture, so I mean, there's no effect because we actually incubated with charcoal strip media. I mean, a charcoal strip serum before, so they weren't exposed to estrogen. And then we actually added only estrogen in.
But can you antagonize some of your data in the female